everybody. So I guess we're going to get started. So my name is Toby Hocking, and I'm a researcher trying to apply supervised machine learning to some big data problems in genomics. So I'm going to start off my presentation with a brief introduction to biology, which is where the data comes from. So I'm going to activate the screen share of this slide, which shows you some karyotype. So this is the chromosomes on the left of a normal human. So you learned in your high school biology class that everybody should have 2 copies of every chromosome. They're numbered from 1 to 22, and then if you're a man, you'll have XY, or if you're a woman, you'll have XX. And so in this talk, I'm going to explain how in cancer we can have copy number changes. So on the right, we see a cancer cell karyotype with 4 copies of chromosome 1, several copies of chromosome 2, and you can have some kind of parts of the chromosomes that are attached to other chromosomes like this. You can have just pieces of certain chromosomes. Just half of a chromosome 5 here is present in half of this blue chromosome is present. And so the data that I will be presenting to you is going to be about trying to figure out how many copies of each chromosome there are in a cancer data sample. So hopefully that's not too complicated, right? So yeah. I ran into these data when I was in my PhD at the Institut Curie in Paris. And so my collaborators were some medical doctors and biologists who are trying to figure out in patients what are the genetic abnormalities which can be associated with bad outcomes. So then they can make better diagnostics and prescribe better treatments to the patients with these kind of cancers. So now I'm going to show you what their diagnosis protocol looked like. So if we look at this picture, we have a figure from one of the papers my co-authors published. So they classified these data into like 2 different categories. So the top category, they classified as like a numerical type of tumor, where you see here on the x-axis we have the different chromosomes 1, 2, 3, 4, all the way up to 22, and then the x and the y. And on the y-axis you have the approximate copy number, right? So in these kind of plots, what we see is that the normal level is in yellow. So this is about two copies, like a normal human cell. And then the gain levels are in red, and then the loss levels are in green. So they classify this profile as a numerical profile because there's only changes in whole chromosomes, right? So here you see the whole chromosome is either normal or gain or loss, right? So the gain is like 3 or more copies of the chromosome, and the loss is probably 1 copy of this chromosome. But what they identified when they were looking at the more violent tumors were that, so you have these segmental profiles where you have some chromosomes, like chromosome 1 and 2 here, which have full chromosome amplifications, like here 3 or 4 copies probably. But then you have some chromosomes, like chromosome 4 here, where you have just a part of the chromosome that is missing, right? So here you see the green dots. This is a loss on chromosome, just the first part of chromosome 4. And here on chromosome 7, they saw that you have a gain and then another gain on chromosome 7. So the goal in analyzing these data is to try to detect these distinct changes, so we can more accurately classify large databases of these kind of profiles automatically. So we can do some kind of diagnosis that says either the kids with cancer have kind of a normal profile, and so it's not a violent kind of cancer, or they have this kind of profile with distinct changes or breakpoints. And then we can classify this as a more advanced tumor, and we can target some more appropriate treatments. Okay, so with that, I'm gonna go back to so yeah. After giving you a brief introduction to these kind of data, now I'm going to show you very quickly the tool that I put together to analyze these kind of data. So as I explained, these guys are just looking at these scatter plots of these noisy data, and they're kind of doing this manual visual classification of each of these profiles. And so there can be hundreds of these things. And so the machine learning problem is then, well, if you give some kind of manual classification for a subset of these profiles, maybe we can train the machine to recognize the pattern that they are looking for. And so then they can automatically go and label many profiles after that. So I'm going to go switch to my SegAnnDB website. So at this point, we have I have my web browser tuned to bioviz.rock.inria.fr. So this is the free demo, live demo website that I put together for this software. So any of you can log on and try the software now if you like. So yeah, so here's another figure, just like the one I showed you, where you have the different chromosomes from left to right labeled by number, and then again we have the approximate copy number signal on the y-axis, and on the x-axis we have the distance along the chromosome. But so yeah, these profiles have not been labeled yet, and so what the website allows you to do, well, first of all, it shows you this kind of default green signal, which is kind of like a default model that is proposed, but you can correct using this interactive labeling software that I developed. So for example, here on chromosome 3, it looks like this is a normal level. And so here is a breakpoint, and so we can add a region on the bottom that says, yes, this is correct. There should be exactly one breakpoint in this region. Here, it seems like that's a loss. So I'm gonna add a blue region that indicates that's a loss region with lower than normal level of copy number. So clicking on chromosome 1, we can zoom in and we can see what this looks like. Well, it looks like here's a region where the baseline level here is normal, so that's fine. And then it looks like over here there's going to be a gain. And over here it looks like there's a breakpoint. We can just add some more labels just to confirm that the model was correct. We can add some a 0 breakpoints region down here to say, well, these 2 points seem to just be outliers, so we don't want to detect that as a real change, just like it did. And then you know, we can zoom in further to see what this region looks like over here on the right. So it looks like this is a change to a loss, and then another change down. So we can say that that's a different copy number by saying it's a deletion, which is even lower than a loss, probably 0 copies of that chromosome. And so yeah, so now we've given a bunch of labels, and so now all of the segments on this profile are labeled now with so yellow for normal, red for gain, and blue for loss. And so this is fine. So now the machine learning problem is well, actually on chromosome 11, maybe we can correct. No, I mean, this looks fine. Looks like maybe this is a gain, and then maybe there's, you know, you can have two gains. So we call this an amplification. This is maybe. Anyway, so yeah, the machine learning problem then is to say, okay, we've got these labels on this one profile. Let's say that we're looking at a completely new profile on an unannotated chromosome, can we come up with a model that is consistent with all of these labels that we just gave, right? Because in fact, I mean, if you go to the homepage of this website that I set up, go to the bottom, it gives you a table of the first few profiles that we have, and then you have this list of all of the 270 profiles. So there's really like hundreds of data sets, one row for each one of these profiles that must be labeled to do the analysis. And so, you know, you could go through and you could give labels all by hand, or, you know, you could use machine learning and see if you can make an automatic prediction on one of these that you don't have enough time to label. Like, for example, in this profile, we have, you know, the automatic predictions given by the model. But the interesting part of this system is, right, so if you see a breakpoint that you don't think should actually be there, you can make it go away. The model should learn and adapt to what your interpretation of these data are. So yeah, that's kind of the basic functionality of the system. And so now I'm going to go. So now I'm going to try to explain a little bit about like what are the algorithms that are underlying the system and why are they interesting with respect to previous work. Right. So let me go back to some visual aids in my slides here. So let's see. Okay, so yeah, so like I said, so this is the way that I framed this problem is kind of a supervised machine learning problem, right? Where you have a doctor, he's going to give some labels about what is the correct interpretation of these profiles in terms of copy number. But in fact, most of the previous methods that attack this problem are completely unsupervised. So on this slide, I give you a list of algorithms since 2004, from 2004 to 2011, that have been proposed to analyze these data, completely ignorant of what the labels are. So I mean, they will give some kind of default guess, like the green model that we saw on the website. But yeah, these previous methods don't really use the labels at all. So they could be arbitrarily inaccurate with respect to what the doctor says is a good interpretation of these data in terms of where the real noise and the real changes are. So the goal of my system was to really come up with a method that could learn and adapt to what the doctor tells you is the real signal and the real noise. So then it can give you accurate predictions, unlike these previous methods, which can be arbitrarily inaccurate. So yeah, so the method that I built off of is this method called maximum likelihood segmentation. So this is a pretty general technique, not at all specific to biology, but really whenever you have some kind of series, it could be a time series or it could be like a series of data points along a chromosome like I have, you can apply these techniques to find where are the most likely changes. So for example, the data that I was analyzing seemed to be approximately normally distributed. Like these approximate copy number measurements seem to be like real numbers. So we applied probably the most basic model, which is the Gaussian maximum likelihood model. And so what this amounts to is an optimization problem trying to minimize the square error between the data Y and the mean level mu. And so that's equivalent to the maximum likelihood Gaussian inference. And so this model is defined for a given number of changes, right? So here we have, I say the y hat k, which is the estimated mean with k segments, is the solution to this minimization problem, subject to the constraint that the mean vector has k minus 1 changes or k distinct segments. So we call this the maximum likelihood segmentation because it is the most likely mean vector, the most likely change points the most likely k minus 1 change points. The only problem with this model is that you have to pick the number of change points. So this is the parameter k, and so basically the learning problem, the prediction problem just amounts to picking what is the right number of changes or the right number of segments. So going forward, so I have some database with labeled profiles here, and I'm just going to explain to you how this model is fit in a very quick geometric explanation. So I'm just going to zoom in on these two profiles, one profile with 0 breakpoints label, and one profile with a 1 breakpoint label. And so for these two signals, we have a sequence, the sequence of maximum likelihood segmentations that we can fit. So for example, here is the maximum likelihood model with 1 segment, right? So this green line is just the mean of all of the data points. That's the maximum likelihood model with no changes. Same thing here for the other profile. So it gets interesting when you go up to 2 segments or 1 change. So this model is the maximum likelihood mean vector with 2 segments. So what happened was we searched for a change after every possible data point, and we picked the one after that gives you the least squared error or the maximum Gaussian likelihood. So again, the mean level here is just computed as the mean of all of the data points, and so this problem can be solved in log linear time in terms of the number of data points using this algorithm called prune dynamic programming by my colleague Guillaume Rigat. So what you can see by looking at these annotated region labels in red and yellow here is that, well, this both of these models are consistent, right? So the model for the top profile has a breakpoint in this region where it says we should have 1 breakpoint, and this profile, it has no breakpoints in the region where we said there should be no breakpoints. So in contrast, we have this model with 1 segment, and so clearly this is not good for this first profile, right? Because this is there should be a change detected, but the green model is not detecting that change, right? So we call that false negative. So going on, you have the model with 3 segments. You can, again, you check for all of the all the models with 3 segments or 2 changes. And the prune dynamic programming algorithm returns that these 2 are the change points that are the most likely. And again, so it seems like it's consistent with the bottom profile because there indeed is the only change points on this profile are here, which are not in the yellow region, so that's fine. But on the top profile, we see that there are 2 changes where there should only be 1. So we call that a false positive, right? So this is the essential idea of the machine learning algorithm. Given these region labels, we can quantify where are the false negatives and where are the false positives. And then we can choose the right number of segments, like for example, 2 segments for both of these profiles here. So yeah, what happens is, so you can compute what is the number of incorrect labels for each of the 2 profiles as a function of the number of segments. So like we saw with the top profile, it's only got no annotation, or no label errors for the model with 2 segments. But for the bottom profile, it seems like there's several profiles with 0 incorrect labels. So what the learning algorithm will do is try to decide between these 5 candidate models, right? So out of these models, which are all equally good in terms of the number of incorrect labels that we have, which one is the most likely number of segments, right? So the other problem that this solves is that the SegAnnDB system I propose solves is the optimal fitting problem. So this problem is quite interesting. So sometimes you get labels from the doctors, right? So for example, these 3 labels here that are not consistent with any of the maximum likelihood models, right? So here is a model with 1 segment, which is clearly not good because we need at least 3 changes here in this profile. With 2 segments, this is a little bit better, only 2 false negatives, right? With 3 segments, this is actually worse because here we have a false positive and 2 false negatives. And with 5 segments, finally, we have these 2 labels which are correct, but this one which is still having a false positive. So in fact, there are no consistent least squares or maximum likelihood segmentation models. So how do we find a consistent model in that case? For this interactive annotation software, it's critical that the doctors will be able to find a consistent model. So how do we provide that to them? So I'm going to briefly explain how we did that. So basically, so yeah, to summarize, there's 4 problems that we solved in this SegAnnDB paper, and basically, yeah, so there's 2 statistical machine learning problems which I just described, right? The first occurs when, so yeah, well, we saw that when there's exactly one consistent model, there's no problem, so we just pick that consistent model and that's fine. But when there's more than one consistent model, we have this prediction problem. Which of the number of change points is the one that we should show? So the solution is we have this learning algorithm that will take all of the other labels from all of the other data sets into account and will give a good prediction about the number of segments that is appropriate for this particular profile. And so the other problem is this fitting problem, where we can, sometimes there are no consistent maximum likelihood segmentation models, but what we did is we developed this specific segmentation algorithm called SegAnnot, which basically takes these annotated region labels into account as a part of the optimization problem. And so it's able to provide a maximum likelihood segmentation no matter what the change point labels that doctors give us. And so the 2 other technical problems that we had to solve to implement this system were these interactive scatter plots where we could zoom and annotate and have the model be updated in real time. So then, and also, we have to store this data in a database so we can learn from it, and also export the data to some genome browsers so then the doctors can view the results of their analysis. So yeah, very briefly about the optimal prediction problem, we have this paper that we published with Guillaume Rigaille in 2010. Well, actually, so it was at NIPS in 2000. Oh no, sorry, it was at ICML in 2011. This paper here, right? So basically, what we're doing is we're solving this maximum likelihood segmentation problem again. And like I said, we can use this print dynamic programming solver, which is implemented in C++. I wrote a Python interface, which is about 50 lines of C code. And so what it gives you is this set of models between 1 and 20 segments, usually. And usually there's not more than about 20 segments in these profiles, so we just fix this as like an upper bound. And so then we have this supervised learning algorithm that basically uses something like linear regression, but with limited labels that we have. We call this interval regression. And so it's going to pick which one of these models is the most likely. And it's going to tell us, okay, we can, which one of these we should display for the doctors. So, and yeah, so to implement this, I mean, basically it's just a gradient descent algorithm. So it's pretty simple, about 100 lines of Python NumPy code. And we just continuously run this on the server as a background process, right? So every few minutes, once you've added more labels, the gradient descent will learn what is the pattern in those labels so it can apply it to future data sets. And so for the optimal fitting algorithm, basically, like I said, we have this SegAnnot algorithm that we propose specifically for these labeled data sets. And so again, it's going to be solving a least squares problem where you're just looking for the mean vector mu, which is closest to your data points y in terms of the squared error. So it's maximum likelihood Gaussian inference again. And so yeah, the difference is just the constraint, which is that the mean vector should have one change in each one of these one breakpoint regions where the doctor specifies there should be one change. So it's a quite straightforward optimization problem. and you can use dynamic programming to implement this. We implemented it in C, and I wrote a Python interface for the website. So yeah, so for these interactive zoomable scatter plots, it's a bit technical, but basically, what we decided to do is, well, first I tried to use D3 by itself, but sometimes there's way more data points than can be possibly displayed in web browsers. So what I did is I said, okay, let's not use D3. This is a very famous JavaScript visualization library. Let's not use it for everything. Let's just use it for displaying the model. So in fact, for displaying the data, what we do is we just display a static PNG image rendered using Python image library. And so this just takes up between 10K and 1 meg of disk space, not a big deal. And it renders quite fast. So yeah, but there's some limit on the size of these. So yeah, I'm not going to go into that. But anyways, so yeah, so the interesting part is that we can interactively add labels and update the model. And so for this, we do render the current regions of the model at inside of an SVG element using D3.js. And so whenever the client, the user of the website, changes the labels, what we have to do is we have to save those labels on the server and then send the new model back to the client for display. So all of this takes about 500 lines of code in JavaScript using the D3 library. And so finally, the other technical challenge in implementing this was to get all of this working together in a database that really we can use supervised learning on. So so yeah, basically, I decided to implement the web server using Python, and yeah, so I used Berkeley DB to store all of the information, because at first I tried something more high level structured, like I think I tried Postgres at first, but to store all of the data and all of the models in one of these relational databases, it seemed like it was way too slow. So we had to do some optimization, and so I chose Berkeley DB because it's very lightweight, very fast, and we can basically store anything. So yeah, and then I chose this Pyramid web framework for gluing everything together, because it seemed like it was very useful for interacting with even databases which are not necessarily relational. So so basically, yeah, I just wanted to mention very quickly, we have this prototype online, we have the source code online. So for future work, what would be nice is we do some kind of active learning strategies. So instead of just asking the doctors to label a bunch of random profiles, what we could do is we could say, okay, label 1, and then we update the model, and then say, okay, here's the next profile that you should label, because this is the one that seems like it will help the model to become more accurate the fastest, right? So this is not something that we have worked on yet, and it would be very interesting for future work. And also the crowdsourcing problem, right? So if everyone, on Amazon Mechanical Turk was logging onto my website and labeling the profiles in a different manner, can we learn a global model that takes everyone into account and gives accurate predictions for everyone? So this is not something that we've considered so far, and in fact, right now we just have frame the problem in terms of, for one doctor, can we give accurate predictions for him or her? Okay, so that's so that's the basic summary. So thanks everyone for listening, and I guess it's time for a question and answer period. So please type some questions if you would like me to answer anything. Did any of you use some kind of change point detection software before? Did any of you try the live demo website that I set up, bioviz.rock.inria.fr? Okay, the question is, any ideas what it cost Stanford to do ImageNet? Yeah, that's a good question. I don't know. So, yeah, creating these labeled databases is something that's quite hard to do without the interactive tools. So, I guess this is one of another contribution of this work that I explained. It makes it quite easy to create these benchmark label databases. So then we can start doing supervised learning and comparing what is the most accurate algorithms for these kind of data. Yeah, there's some idea that maybe using Mechanical Turk to get labeled data will maybe be beneficial. So, yeah, I mean, on the one hand, I think it's an interesting idea from a theoretical standpoint. How do we learn from this crowd of people in a consistent way? But on the other hand, actually, when you talk to these doctors, they do have enough time to label these images. So it's not really a question of whether, you know, we have enough money or enough time to get the labels. It's more of a question of, do we have some software system in place that will make it easy for them to label the things that are obvious for them, right? So, if I understand this question, what is it? Looking at huge images and splitting them into smaller pieces. So I guess with this data, you can do something like this. I mean, all right, we go back to the SegAnnDB website, and I mean, basically we have this, you know, we have these profiles where, you know, I mean, you can just label one individually at a time, right? So you give some labels for this one, you go to the next one, okay, seems like, okay, here's a no change, here's normal, you know. All right, zoom in and now give some labels about where the changes should be. And then move on to some new profile, right? I mean, so there is something like, yeah, you can split the problem into a bunch of manageable subsets and, you know, just do one at a time, right? Yeah, I think the trick is to just, with any kind of machine learning problem, is to try to figure out what is a good abstraction in terms of labels that the experts can provide versus, you know, how hard is it to develop the infrastructure to get those labels, right? So there's no hard and fast answer to these questions yet. It's kind of an art. Some question about many people were using parallel GPU techniques for histology images, brain segmentation. So that's parallel on one image on each GPU core, or I'm not sure. At least for these data with the optimal segmentation algorithm, you have to have all of the data points in memory at a time, right? So if you want to use this optimal segmentation technique to find the most likely breakpoints or the most likely segmentation, then you really have to have all of those data points in memory. So, you know, maybe you can split the image split the data series into a subset and then have a segmentation which is optimal on that subset of data if you want to use the same optimal segmentation techniques. But I guess it's a little bit more complicated for 2D segmentation in brain images, because I guess the dynamic programming algorithm is slower than the log linear time algorithm that we have in the 1D case. Right, the reason that we're able to compute the maximum likelihood segmentation is because we actually have this log linear time algorithm that works that scales with any number of data points. So yeah, it may be portable. This optimal segmentation may be portable from CPU to GPU, but yeah, I think for at least in our context, we have to fit all of the data into memory. So I don't know if that would be a limitation or not. Okay, well, so I guess there's no more questions. So thank you very much for your attention, and I hope you've got some good ideas about optimal segmentation, interactive change point detection, and and have a nice day.